Hi, I am Elaine Seaver. I am a professor of biology here at the Whitney Lab, and I'll be introducing our speaker for tonight. Before I introduce tonight's speaker, I want to encourage you to check out the March Evening at Whitney lecture, which is March 9th, and the title of it is something like How to Build an Animal. So I hope to see you here in a month. It's my great pleasure to introduce tonight's speaker, Dr. Karyn Esser. Dr. Esser is a professor and chair in the Department of Physiology and Aging. This is a newly merged department of these two different areas. Dr. Esser, let me see what I want to say. To tell you a little bit about her academic background, she did her bachelor's degree at Wake Forest, a master's degree at UNLV in Las Vegas, then a PhD in physiology at University of Michigan. After, I like giving the academic history because we all have to move around to the expertise that we're interested in. And so it usually touches on different locations in the country. After professorships at several different locations, she was recruited to the University of Florida in 2015. And she basically, I mean, we're really lucky to have her. She's a highly accomplished researcher in the field of animal physiology, and she's got well over 100, 120, I don't even know, peer-reviewed publications, and she's had ongoing funding from the National Institute of Health for her research. And I'll just point out one thing so I don't go on too long. In 2022, she received the Research Award from the University of Florida College of Medicine, Medicine's Basic Science Research Award for her exceptional research in foundational biology. So I'm very excited to introduce Dr. Esser and hear about her trailblazing research in circadian, the importance of circadian rhythms in cell and muscle health. And before I turn it over to her, I also want to say her work is a great example of the importance of basic research and how you never know when you might stumble on something that could have an important impact in human health. Okay, with that, I will turn it over. Dr. Esser.
Well, thank you for that introduction. That was wonderful, and it's so wonderful to see everybody here tonight. I'm excited to be here. It's great to get out of the swamp and to actually see water and things like that. So yes, what I want to talk to you about today is work that we've been doing over the last 20 some years in circadian clocks and muscle. And I do want to follow up on that comment from earlier about this is accidental. So I did my PhD at the University of Michigan, and it was in muscle physiology. So I was studying a lot of stuff about muscle, but it was in my first academic position at the University of Illinois, Chicago, when we were doing some experiments to try to identify these factors that were regulating gene expression. And we were using some new technology that were allowing us to look at things that we weren't predicting. So sort of what we call unbiased assays. And in looking at muscle, following a bout of contractions, these circadian clock genes came up. And as I was sharing earlier, there's a variety of serendipitous steps that occurred. I mean, I was fascinated with the concept. What's a clock doing in muscle? But there were a series of other things that had to happen, i.e. someone that a senior scientist up at Northwestern that got excited with me and said, wow, you know, we know this goes on in the brain. Why the heck are they going in muscle, you know? And so it to me, it's just like, this is what we're about. We just, you know, this discovery and like, wow, I'm going this way. No, I'm going to go over here and try to figure out what's going on. So I'd like to take you along on the story that sort of has emerged from this. And then I have found, I mean, again, I sort of come from a very fundamental biology background, but I live in the world of the medicine. And so being able to talk to clinicians about, there's some fundamental biology that may affect your patient health, especially, what goes on in an ICU. How do we deal with people in these conditions? And so hopefully I can get some of these concepts across today. So wish me luck. Okay, so I like to start with an outline. There's going to be sort of three main themes that I'm going to try to cover tonight. Let me see if I make sure I push the right button. Okay, so the first one is just introducing all of you to sort of where the field of circadian rhythms is or are. So you can check my grammar. So give you a little background because the field has changed dramatically over the last 5, 10, 15 years. What does a muscle clock do? Why is it there? What is it doing? What happens when we disrupt the clock? And then actually then thinking about, can we actually think about targeting clocks as a therapeutic strategy? And again, I love skeletal muscle. Because I think it's really important. Can we target these muscle clocks actually to improve health? And actually sort of directed toward myself as I age, is this something I can actually think about to try to improve my own health? So I want to start with the concept that virtually all living things on planet Earth have clocks. Okay, so this is a fundamental part of biology. And this was one of the things that actually drew me into this field because I was, again, studying mammals. But once I realized that, let me see, so bacteria have clocks, 24-hour clocks. Plants have clocks. In fact, the very first documented circadian experiment was on a plant where they looked at the leaves opening and closing so they put it in dim light. And it wasn't the sunlight, the actual intrinsic plant was opening and closing. Okay, that was in 1720 something. Insects have clocks, and of course, mammals have clocks. So these clocks have been in all of biology on this planet for a very long time. Now, there are other people that really specialize in the evolution of clocks. I'm not going to get into that, but the fundamental aspect of the clocks is they are based on an approximate 24-hour day. So they're based on the fact that the Earth and the sun have this relationship that's about 24 hours. So again, if there's nothing else you walk away from with this, is that circadian rhythms are just a part of our biology, and it's real, and time matters. And time's going to matter if some of you are on statins. When you take your statin, you should be taking them at nighttime. You can go lower dose, get more efficacy at nighttime. All right. So there are ways in which the time is really going to matter. All right. Why? So who cares? Why do we have circadian rhythms? Well, what we know is that the circadian rhythms are actually there to prepare your body or prepare your cells for expected changes in environment. So again, as I was saying earlier to my host, I could fall flat on my face at this stage of my life. I know the sun's going to come up tomorrow and I will be okay. I may embarrass myself completely in front of you, but I know the rest, life goes on, right? So we all know that there is this 24-hour pattern. The sun comes up, maybe it's cloudy, but we have these diurnal patterns of light, dark, temperature, all that kind of thing. The fact is our biology has evolved under that condition. And so what we now understand is these circadian rhythms, and I will get into the clock part of this, have evolved to prepare us to get ready for these changes in our daily activity patterns, right? Specifically, circadian rhythms contribute to what we call predictive or anticipatory homeostasis. So as a physiologist, maintenance of homeostasis is one of these fundamental aspects. You eat a meal, your pancreas releases insulin, manages glucose uptake. That's what we think of as reactive homeostasis. You eat the meal, etc. What the clocks are doing and what circadian rhythms are doing are actually changing your biology before things happen, so that when you get up in the morning, you're actually ready to go. So in case of my muscle, my metabolism is changing before you wake up. So that if you have to run around and chase down food or find a mate or do whatever, fight off whatever, you're metabolically ready to take that challenge on. Circadian rhythms are important for survival. So there have been experiments in which we can genetically modify the clocks and change the circadian rhythms and then put them in a light dark, so we can make the clocks run at 22 hours. When we put them in 24-hour environment, they don't survive well. We can make them 28 hours. We put them in a 24-hour environment, they don't survive well. Now, if we take these 22 and 28-hour clocks and put them in environments that are either 22 or 28 hours, they do just fine. So what's important is the intrinsic timing of your biology with the extrinsic world that we're living in, okay? So how do we define circadian rhythms? So most of you probably know. I mean, this is not a new term out there. And so we think about circadian biology, and so this is an approximate 24-hour period in which we have events, what did I call them? Processes that occur. So we think of sleep-wake cycles. You can think of changes in blood pressure, heart rate, that kind of thing. But what is critical to this is point 2. You don't need any environmental change, just like that plant. You can put the plant in standard constant light, the leaves will still come up, or you can take a sunflower, the sunflower moves from one side to the other. It doesn't need sun. It does that intrinsically. So our circadian rhythms are actually coming from something inside us and not from, oh, it's sunny out. Okay, so the definition of circadian rhythms are very, very specific to the fact that these are behaviors that occur in the absence of any information from the environment. So those observations in and of themselves have led to the identification that these things are intrinsic. These are intrinsic to our cells, they're intrinsic to our body. And so now we know that circadian rhythms can be detected from the organism level. So we think sleep-wake, we can think, like I said, blood pressure. But actually, did you realize your chromosomes, your DNA is actually opening and closing over a 24-hour period? DNA repair happens over a 24-hour period. All right, so we can go from the very fine molecular level all the way out to the system level, and we can see these patterns every day. So what I'm sharing with you right now is something called an actogram. So as I say, what does it look like to visualize circadian rhythms? So one of the things, like I said, I fell into circadian rhythms. And so one of the things I realized, thank you, before I trip over that again, is that activity patterns are a common way for us to look at circadian rhythms. And circadian biologists have been doing this for decades. And this is an example of this. So this is an example of one mouse. We have it in a cage. It has access to a wheel. And so let me illustrate what this is. So these are the dark cycles. So this mouse is under 12-hour dark, 12 hours light, 12 hours dark, 12 hours light, so forth. And all these little dark tick marks here are cases where the mouse gets on the wheel. Now, mice love to run. And in fact, female mice are better runners than males, just FYI. But so this is probably a female mouse. It's running pretty good. So hopefully what you can see, this is day 1 and day 2. So mice are nocturnal. So they're going to sleep during the light phase. So they're not moving during the light phase. They're getting on the wheel. In fact, as soon as the light goes off, they're on the wheel and they're running. They take a little siesta or snack a little bit here. They get on and then they go back to sleep. Now, again, as I said, circadian rhythms are only defined under constant conditions. This is not constant conditions. This is light dark. So the way we look at circadian behavior is we turn the lights completely off. And so hopefully what you can see, in the absence of any light, these mice are actually quite rhythmic. Now, they're not exactly 24. But they're actually quite rhythmic. So they still get, they figure out at some point, something's telling them, get on the wheel, get on the wheel, get on the wheel, get on the wheel. Now, what we know about this strain of mice is that their internal clock is actually 23.6 hours. So they're slightly shorter than a 24-hour. So each day they wake up half an hour earlier and they just get back on the wheel and they run. So this is how we can look at the intrinsic circadian behavior of mice or of any animals. Is once we put them into constant conditions, we watch for their activity behaviors. Now, I always have to laugh at this because it's sort of like now that I'm doing research in a medical school, working with humans is much more difficult than it was in 1938. So this was the first known circadian experiment with humans, and you can find an article in The Scientist. So this was actually never published in a peer-reviewed journal. So you'll just have to kind of take our word for it. So this graduate student and his advisor, which, go down and basically they want to ask whether there are repeating patterns in their physiology. So they have to go somewhere where they can live, where the light doesn't change and the temperature doesn't change. So they have constant conditions. So what they did is they went, and we actually know who it was. They went down to Mammoth Cave in Kentucky, where within the cave, the temperature is constant. It's at 54 degrees Fahrenheit, and there is absolutely no light. And so what they decided to do in this experiment is they said, okay, let's make a day 28 hours. All right, so they slept for 9 hours, worked for 10, and rested for another 9. And then they monitored temperature and heart rate. And what they found, even though they were living on a 28-hour day, their temperature and their heart rate were still on 24. And so what this said is that, again, it's just that whole idea that it is that intrinsic pattern that's driving these circadian things, and not necessarily light dark, or in this case, there's no light dark. So it's not just I wake up or when I go to sleep. It's actually something within us and within our biology. So blood pressure is another very, very commonly used circadian output. And so I just have an example. This is actually from an older paper in which we have systolic blood pressure. So we have time of day on the x-axis down here. So we're sort of midnight, noon, midnight again. And we've got diastolic blood pressure up here, or sorry, systolic blood pressure up here and diastolic blood pressure here. And I just put in this red line to say, okay, this is when this person was waking up. Hopefully what you can notice is blood pressure is increasing before they ever get up. Again, this is intrinsic. Your body temperature does the same thing. Your heart rate does the same thing. So your heart rate's not increasing, your blood pressure is not increasing because you're getting out of bed and moving. It's actually intrinsically driven. So another example of the circadian system. Did you know that your own strength varies over time of day? So I'm using just one study right here in which I like this study because what they did is they took elite athletes. But there are a number of studies that have looked at, okay, we're going to ask you to do maximum strength measures, at 7 in the morning, and then we're going to do strength measures at 7 in the evening. And, look, I go to the gym. Some days it's like, yeah, okay, I don't really feel like, but we've got elite athletes here. And I don't know if any of you have been around elite athletes, but they're going to take the challenge no matter what. So so we're going to take the motivation out of this assay. And we're going to say, okay, generate maximum strength here. And these are the exact same people at 12 hours later. There's a 12% difference in strength just because of time of day. All right. And so again, we can go through a whole litany of variables in our physiology that exhibit these circadian variations. So where we are with this, circadian rhythms are endogenous to the organism. In the next section, I'm going to talk a bit about the molecular mechanism that underlies this. They can be measured at the behavior level, physiological level, and heart rate, temperature, and blood pressure, things that you get on your wearable devices. And we can measure them at the molecular level. And to define a variable as circadian requires that this pattern has to occur in the absence of environmental cues. So again, light doesn't turn on clocks. light's important for clocks, but it doesn't turn them on. Clocks will always keep running. All right, Nobel Prize. So in 19, God, I'm off by a decade. So 2017, the Nobel Prize in Physiology or Medicine was awarded to these three people: Michael Rosbash, Michael Hall, actually they're all Michaels, and Michael Young. And these guys had identified the molecular mechanism that creates this timer. They did it in flies. They did it in the 1980s. The award was in 2017. So what is the mechanism? So it's a feedback loop, and you don't need to get, I have this simplified cartoon, but there are some molecules in the nucleus. We call them diumal and clock. They bind to pieces of DNA to regulate genes that are part of the period and cryptochrome family. These things get expressed, and in the cytoplasm, these proteins multimerize, and then they get back into the nucleus where they inhibit this. So you have this is a timing mechanism, and it takes approximately 24 hours. So that is the fundamental basis of the timing in your cells. So just to give you a look at, so what does this mean? So I hopefully, let me see if, let's just focus up here on so BMAL1. So this is a gene that is a part of the clock mechanism. And so what people have done in this case, this is from mouse. We've collected tissues every 2 hours over a 48-hour period. And this is why it's really good to be at a good, large undergraduate institution where you can get young people to come in and stay up all night and collect things. And so the PI doesn't have to stay up all night. So anyway, so we collected tissues over all times of day, and then asked, what does the gene expression look like? And I think the main point I want to say is that these things aren't flat, all right? So these things are oscillating. So we have about a 4-fold difference between where the gene peaks at the transition from the dark to the light phase, or in the mouse from the active to the sleep phase, and down to the trough, which is here at the end of the sleep, to the active phase. And so where I came in, so a lot of us just look at gene expression, but the reality is there's this whole temporal dynamic to the gene expression of these clock genes that are based on, the period length is approximately 24 hours. And we think about, again, as we start looking at disease models, as we start looking at aging, this amplitude starts to become important. So unfortunately, as we get older, this amplitude starts coming down. It's not as strong. All right. So I always get the question, because I work, so I actually did my master's degree with humans, and you guys are really hard. Not only that, you're all outbred, so your genetics are really hard. So I actually transitioned into working with animal models, preclinical models. And so we obviously work with mice. So the question is, how does a mouse clock compare to a human clock? Because a mouse is nocturnal, a human is diurnal. And so the beauty of muscle is I can get it from you guys. It's not like your heart or your brain. I can't sample that, but I actually can get repeated biopsies out of your leg muscle. And some people have done that, not me. But and so what we can do is we can actually compare the patterns of the clock genes from humans to mice. And that's what's illustrated here. So we have the one core clock gene BMAL1 in the red, and the other one is the negative limb PER2 in the blue. And I've done this on purpose. So so if we look just at light and dark, they're going to be swapped. But if we think about it as active rest, they're actually exactly the same. So we are not much different than a mouse in terms of our muscle, all right? In terms of the pattern of the clock genes. So you can see during the active phase, the BMAL1 goes up and peaks at that transition to active to rest. And that's true in the mouse as well. And there are a variety of other genes now we've looked at and compared across human and mouse, and there actually is a very strong conservation in pattern. So even though we're diurnal and they're nocturnal, again, if we think of the day as an active versus rest period, we're very, very similar in terms of our muscle clock. So I put this in here. So one of the questions I got when I first started, so I had a number of my colleagues in muscle think I was really, really crazy. And they said, oh, muscle doesn't have clocks. That really doesn't work. And so what we did is we actually, so one of the questions was, does the muscle clock work without the brain? So the brain clock has always been a very dominant feature in circadian biology. So we were fortunate enough, so there is a mouse model. So this is our circadian reporter mouse, in which one of the circadian genes, in this case PER2, has been coupled with the enzyme part of what you see in fireflies, luciferase. So now what we can do is we can take cells out of this animal and we can give them the substrate, luciferin, and every time we see light, that tells us when PER2 is being expressed. So we can actually watch when the clock in real time. So we can do bioluminescence recording. And if this video works, we will, let me see. Now it was working. Oops. I don't know how to get it going on the screen. Do you? Audio visual mistake. But what you've got there is a single fiber on the right. It's a single fiber and a skeletal muscle fiber. Now, one thing I need to note is muscle fibers have lots of nuclei. Each nucleus has a clock mechanism. So what we're going to look at now is light emission in this single cell in a dish, no nerve, no circulation. So what we're looking at is PER2 protein expression over time of day. So hopefully what you can notice is it's oscillating. It doesn't need a system. It doesn't need a nerve. It doesn't need hormones. It doesn't need the brain, right? So this was, and there are a lot of questions that come out of this that are fun to think about. But I think the main point here is the clocks in these peripheral tissues actually work just fine. They don't need any kind of information. This is a fundamental part of who we are. Now, I'm a physiologist, right? So yes, I do some molecular biology, but sort of like, why would I enter into this field? Well, it turns out, so there's a clock that's a timer. So I've talked to you about this part in which these genes regulate a timing mechanism. But why would that matter? Well, it matters because you know what? The clock factors actually regulate lots and lots of genes. So you actually have within every single cell in your body a daily pattern of gene expression that is regulated by this timing mechanism, right? And this is what's critical to link the clock timing to actually the physiology of the cell and physiology of the system. And I'm afraid to tell you, this changes when we age, like everything else. Okay, so every cell has a clock, so lots of clocks in our system. I'm going to the next couple of slides, I just want to talk a little bit about the environment and the alignment of these clocks. Because again, you know, it's just, hopefully it's sort of a clear sense that you want your clocks to be in the same time zone. You don't want your clock and your muscle to be working over here and your clock and your pancreas and so you want everybody in the same time zone. So what do we know about clock organization? So we have this little, you know, we put clocks everywhere. And again, what I always sometimes we'll give talks, it's like, you know, there's a sense that the light turns the clock on. No, we just have to get rid of that. These clocks are going to run. They're just going to run. They don't need the light. But what do they need the light for? Well, the light is important to actually set the phase of the clock. So, you know, we talk about spring forward, fall back, right? So what these environmental cues are going to do is phasing, not turning the clock on or on, but just adjustments, temporal adjustments. So the central clock is in the hypothalamus. and it's in a region called the suprachiasmatic nucleus, or the SCN. And this is where your light cues land. And this is what we know the most about. So we know light comes in, it directly interacts, it goes directly to the SCN, and it makes those little adjustments. All right. And historically, it was believed that the SCN was the dominant over the rest of the body. All right. But we do know that it's very important for things like sleep-wake. So it was in the mid-2000s that we started realizing that, oh my gosh, there's these clocks in every cell of the body, virtually every single cell of the body. And over the last 10, 15 years, we've learned that things like, what time do you eat? What time do you stop eating? When do you exercise? These things all provide information to clocks in your periphery in terms of their setting, independent of what the brain's doing. Okay, so how you live your life in terms of feeding, in terms of physical activity, these things all provide information to the clocks and manage their settings. Okay. So how much does the SCN control the pace of the clock? It's not completely clear, but we do know. We can take animals and keep them on normal light-dark cycles and feed them in a different, or we exercise them in a different part of the day, and we can disconnect the clocks. So the SCN will stay attached to whatever the light-dark cycle is, but if we eat in the middle of the night, our liver clock, our muscle clocks, our heart clocks, they're going to start moving to when we're eating. So the SCN is not dominant in this case. It's more of a conductor, but it can't control everything. So that's what we say, is think of the SCN clock more as a conductor rather than a drill sergeant. So jet lag. I assume most of you have experienced jet lag at some point in your life, or even that discomfort that you have with particularly spring forward. So there is a difference between going west to east or east, you know. So spring forward is more difficult for our biology than fall back. But let's say we travel, and so I'm in Gainesville, let's say I travel to Europe, so I have a 6-hour shift. I'm not going to feel great for a while. All that, what that is circadian misalignment. Obviously, it's not a disease, it's a problem with our clocks. Our clocks are not aligned. And we now know there's something called, did I put it here? Maybe, oh, I must have put it somewhere else. But there's also what we call now, because we can eat anytime, we can do social jet lag. So you too can feel jet lag without leaving your home, just by screwing up your feeding pattern and your lighting pattern. Oh, that's, yeah. So basically, this has become a significant aspect. So it's, you know, things like light at night. So, and when we get into light, then you get into wavelength. Is it blue light? You know, that kind of thing. But but the feeding thing is a big deal. And for some of you maybe have heard of, there's a lot of people in the intermittent fasting world right now. And its health benefits. Now it's a little messy. What we are arguing, both within my lab and within a group of us at Florida and elsewhere, is that the timing of feeding is actually going to be important. And we're hoping to start some clinical trials in this soon, because we see this as an important time cue to consolidate clocks during the time you want them to be there. All right, so we'll move sort of quickly through the muscle clock. So if the muscle circadian clock is important for physiology, what happens when we stop it? So again, I work in mice because there's a lot of things you can control in mice that you can't do with humans. And one of them is we can actually genetically modify the clock. And so all this slide here is to demonstrate is that what we did is we took a mouse and we're going to go and we're going to stop the clock in an adult. We're not going to affect the development. We're not going to affect that. We're going to wait until they're an adult and we're going to stop the clock only in the muscle. Brain clock, all the other clocks are just fine. What happens? They get weak. So one of the things that's really nice with mice is they'll grab onto things. So we can do what's called grip strength, and that's sort of illustrated up here. So you can hold on to them. They'll grab onto this wire mesh, which has a force transducer, and we can measure grip strength. So what we see with these mice is they've got about a 20% decline in forelimb grip strength. Now, again, I'm sort of a muscle purist. This is a neuromuscular. It's got to take the brain, a variety of things. So then when we get to the very, very fundamental assay of what does the muscle do? So what we can do, I don't know if you can see this little pink tissue right here, but we can actually take the muscle out of the animal, stick it in a bath, give it all the, oxygen and glucose and all the things it needs. And then we can artificially stimulate it to contract. We can do artificial innervation, and then we can measure force output, and we can normalize it for size. And so as you start increasing the frequency of stimulation, you start increasing the muscle force, and you reach a maximum. What we find in this muscle that has a stopped clock is it is weaker. It's just intrinsically, it's not the nerve, it's not anything else, it's intrinsically weaker. It can't generate the same amount of force. So we have a weaker muscle with these animals. Now, probably more clinically relevant is what we found is there's something going on with glucose metabolism in the animal. So in this case, so we've got these same animals. So again, we've got the normal mice, and now we're going to measure blood glucose in our muscle-specific knockouts. There's no difference. But fortunately, I had a graduate student that was smarter than me and said, okay, well, I'm going to take the blood and measure the insulin levels. And what she reported was like, oh my God, to maintain that normal glucose, we needed almost 4 times more insulin in those mice. So that was our first clue that something is happening systemically in terms of glucose homeostasis. So then we did a variation of a glucose tolerance test. So again, we just squirt some glucose into the mice. And in this case, we're going to monitor every 15 minutes. We're going to measure blood glucose. So here's the normal excursion. So we put glucose in their bellies. We see the increase in the blood. And then it comes down within, 45 minutes to levels that are not very different from control. In contrast, our mice that are lacking the clock only in muscle, not pancreas, not any of the other tissue. The excursion is for the same amount of glucose. This excursion is significantly higher, and it takes longer to recover. And so the way in the field we look at that is sort of area under the curve. And as you can see, we basically have a pre-diabetic mouse, just by stopping the clock in muscle. I'm going to pass this. So what we did find out is that these muscles are no longer insulin sensitive. Okay, so insulin normally will increase muscle glucose uptake. It doesn't work anymore. This one, I remember sharing this with some freshmen at UF, and they kind of scared them a little bit. But what we did is we took some of these mice in which the muscle clock is disrupted, and we said, okay, let's stress them a little bit. So what we did is every week we shifted them by 6 hours. So basically thinking about taking you from here, flying you to Paris each week, all right? And people had done this with aging mice, and they found, unfortunately, that aging mice start dying. Well, our mice are starting to die. So by changing the muscle clock, we have altered the systemic health of these mice so that now when we stress them, I mean, we're not exposing them to COVID. We're not exposing them to some kind of cardiovascular event. We're just shifting the light phase. We're making them much more vulnerable to some kind of stress. All right, so stopping the muscle clock leads to loss of homeostasis with decreased muscle function and metabolism, and we have a more vulnerable mouse. So I did want to transition. So we've got a few minutes left to some of the work that we're doing in terms of can we actually rescue some of these problems. So this was a study that was done very recently. We just published this work with a great team of colleagues at the University of Florida. Just asking a very simple question. What happens to clock function as the mouse ages? So we worked with 3 different aged mice, and these are the human age equivalents, 25, 60, and 90. We collected tissues, and we collected muscle, we collected heart, brain, lung, kidney. And over this 48-hour period, and then we used RNA sequencing to say, okay, what's happening to what we call the clock output? So what you're looking at here, so again, if you think back to my earlier slide about how the clock regulates a gene expression program. So for example, in a young muscle, there are about 940 genes that are regulated in a daily pattern. So this is a heat map. So here's where they're peaking, here's where they're troughing. All right, as you get to sort of middle-aged, you're down to 699. And now by the time you're 90 years old, you're 474. If some of you think about patterns, hopefully you'll notice this is a nice diagonal. We start getting very, a little bit more rigid. And by the time you get to this very old age, it's kind of unimodal, I'll call it. Now, so what we do know is the output from the clock is declining with age. And in fact, so that what this Venn diagram tells us is there are actually very few genes that are actually maintained. So when we think about, so the genes that are being regulated here are regulating all kinds of very fundamental things like protein turnover and insulin sensitivity and all these sort of what we would call housekeeping things. By the time we get out here, they're just like, help, I'm just trying to respond and just do what I can do to stay alive, all right? So what we're learning from this is the clock is ticking, but it's just actually not, it's the way I think about it is this thing's turning over, but the gears aren't engaging, right? So it's slipping and it gets a little bit more slippery as we get older. So can we use genetics to actually try to rescue this? So what we took advantage of for this is that the mouse that lacks BML in all the cells is actually now considered a model of advanced aging. So in particular, these mice die at about 38 weeks of age, which is about a 40-year-old lifespan. So this is work from a postdoc in the lab, Miguel Gutierrez Monreal. So what he did is he took these BML knockout pups. We used AAV technologies to introduce this clock gene. Now we're reintroducing the clock gene back into the mice, but we've done it in a way that it will only be expressed in skeletal muscle. So we're going to do it because, again, you've got muscle everywhere, right? You got your diaphragm, you got muscle up in your jaw, you got muscle down here. So we can't just do one injection. We have to do systemically and we have to control it. So all this slide is here to say is that when we did this using the AAV and we put BML back into muscle, what we did see is we actually restored or rescued normal clock gene expression. So one of the questions we didn't know. So this is an animal that doesn't have a functional clock. Can we actually put a piece back in and actually get the clock back ticking again? So what this first data was really important for us to demonstrate that, yes, in fact, you can start with a cell that doesn't have a clock, it's missing a piece. We can add the piece back in, and that is sufficient to get it ticking again. But I do want to point out here is that, the technology has its limits. I mean, there's AAV technology is wonderful, but we actually only rescued about 30% of normal expression. So one of the questions was, what happens to the muscle? And so these are just some cross sections of muscle fibers. So here's a normal healthy muscle. This is the knockout muscle. This is the knockout with AAV. And what a lot of people, so one of the things we know is that in the knockout animal, the muscle fibers are smaller. So if you actually look at the size, cross-sectional area of this muscle, it's actually bigger here than it is here. When we rescued with AAV, we did absolutely nothing to muscle size. So we're not making these animals buff. They're not going to, they're little scrawny little things, okay? So now we go back to an assay that has been around for hundreds of years and ask about muscle function, but we do rescue muscle force, okay? So what we're doing, so even though we're not rescuing size, we're making the muscle generate the force that it normally would. So that's a good thing. So what systemic parameters are changed? Body weight, So there's a lot of data here. Miguel would go down every week and weigh them. And so what you see is these knockout animals they just don't gain weight. So here's a normal mouse in the black boxes. So and these are all males, by the way, right now. And so what you see is they look pretty much similar through 12, 14 weeks, but the normal mice continue to gain weight. Our BML knockouts or the ones with AAVs, they're not going to change body mass. So we didn't do anything there. We looked at food intake. We also did body composition, so percent lean, percent fat, no difference. But we did rescue so just like sort of the reverse of the knockout. So what we see is the knockout mice have higher blood glucose and longer to come down. So that's the red here. So they're going way up, they're coming down. So the wild types are here in the middle, and we've actually improved glucose tolerance, again, just by targeting the muscle. So remember your muscle. And so that's what this is, just again, that area under the curve. So we've now restored that glucose tolerance to something like the normal muscle. Activity patterns, right? So these are the actograms. These are just example actograms. And really, it's not so much here because, so these mice, again, remember, they're not no clock anywhere except for clock rescue in the muscle. The animals are still arrhythmic. They don't know, they don't have that normal sort of 24-hour pattern of activity behavior. So the muscle's not governing that. But what we did notice is, and again, this is voluntary wheel. This is not your personal trainer taking you and say, get on the treadmill. You know, they voluntarily were more active. So just making the muscle healthier actually increase the activity pattern of these animals. And I think there's we're starting to do some brain muscle interactions. How does the healthy muscle talk to the brain in terms of affecting voluntary activity? Here's the whole thing. We increase longevity with these mice. All right. So here's the knockout mice. And we get 50% survival, median survival, 38 weeks, just like all the published. So I can say we're reproducible here. We can do that. When we restore BML only in muscle, we actually have a significant increase in the longevity of these mice. And remember, this is just 30%. We haven't fully restored. So one of the experiments we have now are using some new techniques to increase the expression of the clock to see if we can actually improve this. And the good news is the trend is there for females as well. This seems to be both common in males and female mice. So to finish off, circadian clocks are important for health. These cell clocks tick, but they also get information that controls their settings from how we live our life, what time we eat, what time we exercise, how much light we get exposed to at different times of day. Skeletal muscles have clocks. And when the muscle clock doesn't work well, muscle strength and muscle metabolism decline. And so there are, you know, again, we think about Olympic athletes. If they're going to go compete, let's say in Beijing or in some other place. And so the athletes already know this because they get to wherever they're going to do their event, they get there at least 2 to 3 weeks ahead of time. You know, some of it may be adjusting to food, but the reality is it's about adjusting the clocks so their performance is improved. Muscle clocks can be rebooted. So we can use genetic approaches. Again, we're not really thinking about doing that to humans, but we are thinking about, can we actually do some lifestyle interventions like time of feeding, time of exercise, to improve clock function in peripheral tissues as well as the brain. And so this is an area that we're really excited about across the department. Obviously, I'm personally very excited about this, but can we use this? Because we also see clock disruptions in cancer and muscle. A variety, type 2 diabetes, clocks are disrupted. Almost all chronic diseases, we see clocks disrupted. So can we actually start using, you know, so yes, there are some drug development coming along. But even in the short term, we can think about what are we doing with our lifestyle in terms of when do we eat, when do we get sun, and when are we active? And can we use these kinds of interventions to at least help the system help itself? Okay. And last but not least, I have a wonderful group. I've really, I'm not a native Floridian, more of a Chicagoan by nature, but it's been a wonderful time at the University of Florida. They've provided a fantastic environment. And obviously all the people that do the work are the ones that really deserve the credit. I get the fun of coming out here and talking to you. So thank you very much.